Hello everyone, I'm Dr. Lowe-Power, but you can call me Tiffany. I'm going to give a short introduction to our lab, and I first want to start with introducing our physical space, because as a new assistant professor, the newest lab in the department, this is something that we've been putting together over the pandemic. So we're really proud of how many boxes we've unpacked. And how organized we have gotten the space. So we've got room for 6 full-time workers. Currently don't have that many people hired. And yeah, so we're a bacteriology lab. We study the genetic determinants that help bacteria colonize plants and also survive in the environment. Our favorite organism is Ralstonia solanacearum, which causes wilting diseases of many plants. And I've got some Ralstonia on my postdoc's plates, not actually mine. It makes these really beautiful goopy colonies. They'll actually drip onto the lids if you have them inverted, so they're really gross. And yeah, we use a lot of different genetic methods in the lab. We are really interested in mutant phenotypes. We've started using a technique called random barcoded transposon site sequencing, which allows us to measure the phenotypes of thousands of mutants at once in a single experimental condition. So one big thing that we're doing in the lab is seeing which genes are required for fitness at each stage of the life cycle of Ralstonia. And one of the projects that we're working on was actually kickstarted by master's student in the lab, Jonathan Beutler. And this is his bench. Unfortunately, I cannot introduce you to him because it's a pandemic and we're working shifts. But Jonathan's work has been characterizing the virulence of 19 different strains of Ralstonia. They're all really closely related, but from prior ecological work by Philippe Crior's lab, we know that the strains in this group have varying host range. So even though they're quite closely related, they have enough genetic differences that allow them to be very aggressive on melon, or other strains are very aggressive on banana. And so Jonathan's doing all the plant virulence assays. So one project that we're going to be is going to be open in the lab is going to be sequencing the genomes of all these strains and seeing which genes correlate with high virulence on melon and high virulence on banana. Additionally, we have some of those mutant technologies in one of the melon aggressive and one of the banana aggressive strains. So you could also do high throughput mutant screens to see which genes are required in different hosts. So yeah, that's probably the best introduction to the lab. Personally, I've been spending the day running around the lab, reorganizing it, trying to figure out the final home of where all of our equipment's going to be. So that's a bit why I'm a little bit disorganized myself right now. But I hope you guys have a nice virtual visit and you know, get a chance to see whether you'd like to do your PhDs at UC Davis. We'll be happy to have you if you are happy to come. So take care, everyone.